So we will discuss the questions chapter wise. We will discuss where you can expect questions. Clear. So the expected area we will discuss where you can get more questions. So what are the areas in each topics you have to cover in depth. So we can start from topic to topic. Okay. So first one related to epithelial tissues, or this chapter is structural organization organisms. Here you can see different types of tissues are there. In these tissues, when you are studying the tissues, you have to focus on epithelial tissue, connective tissue, muscular tissue, and nervous tissue. Here you can see when you are studying the epithelial tissue, you have to focus on the location. For example, this question is related to location. Columnar epithelium, where it is present, columnar epithelium. Columnar epithelium is present in the absorptive surface. So you can see the columnar epithelium is present in the lining of stomach and intestine. Ciliated epithelium, cilia is present. It helps in the removing of the dust particle that present in the inner lining of bronchioles. Squamous epithelium is lining the blood vessels, lining the lymph vessels. Glomerulus, it is present. Bowman's capsule. Okay, so this squamous epithelium, you can say that it is present in the endothelium. Cuboid epithelium. Cuboid epithelium is present in the duct of salivary gland, duct of pancreas, tubules of nephron. So you can see this duct of glands. Okay, in this way you can answer these questions. Next one, bronchioles. Bronchiole is present in the, where it is present, bronchioles present. Ciliated epithelium is present in bronchioles. Goblet cells. Goblet cells are unicellular columnar cells and it is acting as a unicellular gland. So you can see glandular tissue. Tendons. Tendons are a particular type of dense, regular connective tissue. In this, fibers and cells are arranged in a particular fashion. That's why we will call it as dense, regular connective tissue. Clear? Adipose connective tissue. Adipose and areolar connective tissue both are loose connective tissue. Clear? So this match the following type of questions you can expect from this part. Goblet cells. Goblet cells of alimentary canal are modified from where it is modified. Goblet cells are unicellular columnar cells. Okay, columnar epithelial cells. These goblet cells will produce mucus. So mucus is the secretion of goblet cell, and this mucus is glycoprotein in nature. Glycoprotein. So chemical nature of mucus is glycoprotein, and it is unicellular. Which epithelial cell from columnar cells it is derived? Next question. In both sexes of cockroach, a pair of jointed filamentous structure called anal cerci. Anal cerci is present in both male and female cockroach. Okay. So which segment it is present? 8th and 9th segment, 11th segment, 5th segment, 10th segment. It is present in the 10th segment. But you can see the anal styles. Anal style is present in the 9th abdominal segment. So it is present only in case of male cockroach. Okay. So in this way we can distinguish between male and female cockroach. Match the list 1 and list 2 related to the digestive system of cockroach. So here these are the things related to the digestive system. The structure used for storing the food. Storing the food, we can say crop is used for storing the food. Ring of 6 to 8 blind tubules at junction of foregut and midgut. Between foregut and midgut, we can see the gastric caecae. Ring of 10 to 150 yellow colored thin filamentous at junction of midgut and hindgut. So midgut and hindgut, we have this cockroach is having Malpighian tubes. Structure used for grinding. Grinding structure is gizzard. Gizzard is the grinding structure. Clear? So in this way, you can rearrange the things. Foregut and midgut, gastric caecae is present. Midgut and hindgut, Malpighian tubule is present. Which of the following is the characteristic feature of cockroach regarding sexual dimorphism? So sexual dimorphism, in what basis we are classifying the male and female cockroach? Here some options are given. Presence of anal cerci, that is present in both male and female cockroach. Dark brown body color and anal cerci. presence of anal styles. This anal style is only present in male cockroach. So in this way we can distinguish male and female cockroach. In cockroach, excretion is brought about by phallic gland, uricose gland, nephrocytes, fat bodies, collateral gland. Main excretory organ in cockroach is malpighian tubule. In addition to that, there are additional structures. For example, uricose gland is required for the excretion purpose, nephrocytes, fat bodies. So these all are required for the excretion process. Collateral gland is producing the ootheca. Ootheca is produced by collateral gland. Phallic gland helps in the formation of spermatophore, packets of spermatophore, packets of sperm, that is known as spermatophore. So the covering of spermatophore is produced by this phallic gland. Next one, match the following columns with reference to cockroach and select the correct one. Grinding of the food particle. So here grinding purpose, gizzard or proventriculus. Secrete gastric juice. Hepatic CK is secreting the gastric juice. 10 pairs of spiracle is present in cockroach. Anal cells is present in 10th segment. Anal style is present in 9th segment. So in this way we can match the things. Now select the correct sequence of organs in the alimentary canal of cockroach, starting from mouth. Starting from mouth, after the mouth, cockroach having pharynx, correct. Cockroach having esophagus, that is also correct. Store the food in crop. After that, grinding the food, correct. Ileum, colon, C. Which of the following feature is used to identify male cockroach from a female cockroach? Presence of boat-shaped sternum. Boat-shaped sternum is not present in the 9th segment. It is present in the 7th segment, abdominal segment. Presence of caudal style. Caudal style is also known as anal styles. Forewings wings, darker segment. Presence of anal cerci. Anal cerci is present in both male and female. So the caudal style, anal styles are the things that present only in male cockroach. In male cockroach, sperms are stored in which part of the reproductive system? Testis is producing the sperm, transport by vas deferens. So in this, seminal vesicle is used for storing the sperm in case of cockroach, but in case of human beings it is not used for storing the sperm. The body cells in cockroach discharge their nitrogenous waste. Nitrogenous waste in cockroach is uric acid. Nitrogenous waste, uric acid. In hemolymph, mainly in the form of which form it is eliminating? It is eliminated in the form of potassium urate. Next one, select the correct route for the passage of sperm in male frog. So sperm is produced in which part? In the sex organ. That sex organ we call it as testis. So testis is connected to, it is not directly connected to the Bidder's canal. It is connected to the vasa efferentia. Then it is entering into the kidney. So in kidney, Bidder's canal is there. Bidder's canal is connected to the urinogenital duct. So in male frog, this ureter is acting as a urinogenital duct, both for the transport of sperm and also for the urine. Then last, it is connected with the cloaca. So cloaca is the chamber which is receiving the duct from the digestive system and urinogenital system in case of male frog. So this option is correct. So here you can expect the question related to this digestive system, reproductive system. What are the structures present in the reproductive system? So focus on these points. Frog's heart, when taken out of the body, continues to beat for some time. So continuous to beat means there is a property, that is auto excitability is there. That's why the frog heart is continuously beating. In our case also continuously, our heart also continuously will beat. So frog is poikilotherm. What do you mean by poikilotherm? Poikilotherm means body temperature will change with the environment temperature. So this is not correct. Frog does not have any coronary. So coronary circulation is not connection with this. This is regarding the beating of the heart. Heart is myogenic and auto excitable. So these two things are correct. Now the chemical analysis, biomolecules. Lipids have only single bond are known as unsaturated fatty acids. So in unsaturated fatty acids, there will be double bond between the carbon atom. Okay. So that type of lipids contain unsaturated fatty acid, and these are unsaturated one. Here single bond is present, so it is not correct. Lecithin is a phospholipid. Lecithin or phosphatidylcholine. That is a phospholipid. Correct. Trihydroxypropane. Glycerol is trihydroxypropane. That is also correct. Palmitic acid has 20 carbon. Palmitic acid has 16 carbon. 16 carbon is palmitic acid. 18 carbon is stearic acid. 18 carbon is oleic acid. 18 carbon is linoleic acid. Linolenic acid. Arachidonic acid have 20 carbon, not 16 carbon. So these two options are correct. Identify the basic amino acid from the following. Glutamic acid. Glutamic acid itself is a acidic amino acid. Lysine. Histidine, lysine, and arginine. These are the basic amino acids. Arginine. These are basic amino acids. Okay. So this is lysine is there, so lysine is correct. Valine is neutral amino acid. Valine is a type of neutral amino acid. Category is neutral amino acid. So you can expect this question which comes under neutral amino acid. Valine is neutral amino acid. Tyrosine is non-essential amino acid, and tyrosine is also aromatic amino acids. From the tyrosine, epinephrine, adrenaline, noradrenaline, T3, T4, triiodothyronine, and tetraiodothyronine. These all are found from tyrosine. So tyrosine is aromatic, and also tyrosine is non-essential. Okay, these are the features. Lysine is the correct answer here. Typical fat molecule. So in a fat molecule, what is there in the fat molecule? In a simple fat, you can see there will be glycerol, CH2OH, CHOH, then CH2OH. So this is the glycerol. Glycerol will be attached with fatty acids. So there will be, if there is one fatty acid is there attached to the glycerol molecule by forming ester bond, we will call it as monoglyceride. Then diglyceride. One more fatty acid, if it is there, diglyceride. If it is one more is there, then it is known as triglyceride. So one glycerol attached with three fatty acid molecule. So that is the simple fat. Okay, fat molecule. Which of the following is not a secondary? This is very important question. Secondary metabolite, which is not a secondary metabolite. Curcumin is a secondary metabolite, so it comes under drugs category. Vinblastine, turmeric. Okay. Morphine is an alkaloid. Anthocyanin is pigment. Lecithin. Lecithin is what? Lecithin is phosphatidylcholine, or it is also act as a surfactant. Lecithin is also act as a surfactant. So here the lecithin is the incorrect one. Lectin is what? Lectin is a secondary metabolite, but lecithin is not a secondary metabolite. Lectin is a secondary metabolite. Now we can categorize the thing, match the list. Terpenoids. Terpenoids here diterpene. Diterpenes comes under the category of secondary metabolites, that is terpenoids. Lectins. Lectins are concavalin A is an example for lectin. Alkaloids. Alkaloid. Alkaloid. This is alkaloid. Codeine is an alkaloid. Morphine is alkaloid. Morphine is extracted from Papaver somniferum. Toxin. Abrin and resin. These are toxins. Clear. So in this way you can expect the question. Secondary metabolite is one of the important part in the biomolecules. Inhibition of succinic dehydrogenase enzyme by malonate. Malonate is inhibiting this enzyme. Is a classical example of. So here malonate is competing with the succinate for the active site of succinic dehydrogenase. So the type of inhibition is competitive inhibition. So in competitive inhibition, Km Km value. What about the Km value of competitive inhibition? Substrate concentration at which half Vmax is attained. Km value and Km value will increase. Then Vmax remains same. Okay, next one. Which of the following describes the given graph directly? So here you can see this graph, which type of reaction is exothermic or endothermic. So here substrate energy, you can see potential energy of the substrate is more compared to the product. So substrate is more, product is less, energy is lost here. So this is exothermic reaction. Exothermic. Exothermic reaction. Okay. Now this is regarding the reaction where the enzyme is present. So it is decreasing the activation energy. So if enzyme is involved, activation energy will decrease, then more substrate can participate. Okay, so here activation energy is more, here enzyme is not involved. So it's an exothermic reaction. This is A is with the presence of enzyme and B is with the absence of the enzyme. Option D is correct. Exothermic reaction with energy A in presence of enzyme. So enzyme is decreasing the activation energy so that more substrate molecules can participate in the chemical reaction. Which of the following statement is incorrect? Incorrect statement. The competitive inhibitor does not affect the rate of breakdown of enzyme substrate complex. That will affect. Presence of competitive inhibitor decrease the Km value. So if the competitive inhibitor is there, more substrate has to be added to reach half Vmax. So we have to add more substrate to reach the half Vmax. So Km value will increase, not decrease. Okay. Competitive inhibitor reacts with the enzyme form enzyme inhibitor complex. That is correct. Competitive inhibition, the inhibitor molecule is not chemically changed. Inhibitor molecule will not change chemically because why? Because here enzyme and inhibitor not chemically reacting. So this is the incorrect statement. Next one. Select the option which is not correct with respect to enzyme action. Which is not correct with respect to enzyme action? Malone, it is a competitive inhibitor of succinic dehydrogenase. Substrate bind with the enzyme at active site. Addition of a lot of succinate does not reverse the inhibition of succinic dehydrogenase. So here it will reverse the inhibition, because actually what we are doing here, we are diluting the presence of inhibitor by adding more succinate. So more substrate is adding and diluting the inhibitor. Clear? So it can reverse the inhibition by adding more substrate. So which is the not correct one? So this is the not correct. Tidal volume. Tidal volume means what? Tidal volume is the volume of air a person is taking in and gives out during the normal inspiration expiration process. That's around 500 ml. 500 ml is the tidal volume. So a normal breathing, how much volume of air the person is taking in or the person is giving out, that we call it as tidal volume. Okay. And expiratory reserve volume is given. Expiratory reserve volume. Expiratory reserve volume is 1000 ml. 1000 ml is the expiratory reserve volume. What will be his expiratory capacity if the residual volume is 1200? Expiratory capacity, how we will measure? Expiratory capacity is equal to tidal volume plus expiratory reserve volume. So tidal volume is given 500 plus 1000. Clear? 1500 ml. Okay, next one. Total lung capacity is the total volume of air accommodated in the lungs at the end of forced inspiration. This includes total lung capacity we cannot measure using the spirometer, Wherever there is a residual volume is involved, that cannot be measured using the spirometer. So total lung capacity is equal to tidal volume plus expiratory reserve volume plus inspiratory reserve volume. Okay, this is the total lung capacity. Residual volume is also included. So up to this we can say, what is this? Up to this is the vital capacity. Vital capacity plus residual volume. That we can mention. So residual volume, expiratory reserve volume, tidal volume, inspiratory reserve. So this is the correct one. Next one. Select the favorable condition required for the formation of oxyhemoglobin. So oxyhemoglobin is formed in alveolar surface. In alveoli, oxyhemoglobin formation takes place. So in alveoli, the partial pressure of oxygen will be high. Partial pressure of oxygen will be high. Partial pressure of carbon dioxide will be less. If the partial pressure of carbon dioxide is less, what will happen? Hydrogen ion concentration also will be less, because here hydrogen ion is forming by dissociating the bicarbonate. So in this way it is not formed. So here option is high partial pressure of oxygen, low partial pressure of carbon dioxide, less hydrogen and lower temperature. So this is the required condition for the formation of oxyhemoglobin. Reverse process will take place in the tissues. For the dissociation, what is required for the dissociation? This is regarding the association. Okay, for dissociation, low pressure of oxygen, high pressure of carbon dioxide, high hydrogen and high temperature. This takes place in the tissues, dissociation process. So association is taking place in the alveoli. These are the factors required for the association. Low temperature is also required for association. The reverse process will take place in the tissues, that is dissociation. Yeah. Partial pressure of oxygen and carbon dioxide at alveoli. Partial pressure of oxygen. Partial pressure of oxygen is 104 mmHg. So this is the partial pressure of oxygen at alveoli. In atmosphere 159. Partial pressure of carbon dioxide is 40 mmHg. Partial pressure of oxygen in this aorta and pulmonary arc veins will be 95 mmHg. Pulmonary vein contain oxygenated blood, aorta also contain oxygenated blood, artery also contain. So in all these it will be 95 mmHg. So in tissues, carbon dioxide concentration. In tissues, what will be the carbon dioxide? It will be 45. In deoxygenated blood also it will be 45. Okay. But oxygenated blood it will be 40. So you can expect the values regarding the partial pressure of oxygen in different blood vessels, in tissues, in alveoli. So this transport of oxygen takes place, transport of oxygen or carbon dioxide takes place from high pressure to low pressure. There should be a there should be a pressure gradient for this transport. Now the question related to the functions. Pneumotaxic center, where it is located? Pneumotaxic is located in the pons region. So here it is controlling the breathing process. How it is controlling? This pons is controlling the, or it can increase the breathing rate by reducing the duration of inhalation. So reducing the duration of inhalation and increasing the breathing rate, that's a function of pons. So pneumotaxic center is located in the pons. Oxygen dissociation curve. So it is actually defining the affinity of hemoglobin and oxygen. See, this adult hemoglobin, and if you are comparing the fetal hemoglobin, the affinity will be more in case of fetal hemoglobin compared to the adult. So if you are drawing the oxygen dissociation curve, there will be left shift and there will be right shift. So where you can see the left shift, if the affinity is more, and the curve will shift towards the left. If the affinity is less, it will shift towards the right. So in tissues, the oxygen dissociation curve is shifting towards right. What does it indicate? Right shift is indicate dissociation and left shift is indicating the association. So if it is more left, it indicates that there is more affinity between hemoglobin and oxygen. Carbonic anhydrase is an enzyme, is present in RBC and also in plasma. So here RBC is given, so we can mention RBC. Primary site of exchange of gases is alveoli. Alveoli is given. Gaseous exchange takes place in the alveoli. So why there is gaseous exchange in alveoli? Because alveolar membrane is very thin and it is made up of simple squamous epithelium. Now question regarding the bloods. Basophils are most abundant WBC? No. Basophil is not abundant. Basophils secrete histamine, serotonin, and heparin. So these three chemicals are secreted by basophil, and the same chemical will be secreted by the mast cell also. So mast cell and basophil both are the same in the secretion of these chemicals. Histamine is a vasodilator. It will dilate the blood vessel. During the allergic response also, blood vessel dilation takes place due to the production of histamine by mast cell. Serotonin will constrict the blood vessel, so the blood pressure increase. Heparin is a natural anticoagulant. So this is true. Basophils are involved in inflammatory response. Correct. Basophils have kidney-shaped nucleus. Basophils having S-shaped nucleus, not kidney-shaped. So this is not correct. Basophils are agranulocytes. Basophils are granulocytes. So these two things are correct. Next one. Arrange the following formed elements. Formed elements in blood cells in the decreasing order of their abundance. Which is more in our body? Erythrocyte is abundant. 5 to 5.5 million is erythrocyte. After that, there are platelets are there, 1,50,000 to 3,50,000. Then we have WBC. So here, which is the WBC? Neutrophil is more. 65% is neutrophil. 65% is neutrophil. 6 to 8% is monocyte. The same 6 to 8% is the percentage of blood plasma protein. So blood plasma protein percentage and monocyte percentage is the same. 3 to 4% will be the eosinophil. So this is the decreasing order. Increasing order, so you have to remember the values, which is more, which is less, so you can identify these things. Person with AB blood group. AB blood group means what? AB blood group means antigen A and B are present on the surface of the RBC. Consider this is the A antigen, this is the B antigen. In plasma, there will be no antibodies against this antigen, so that AB blood group can receive all types of bloods. Absence of antibodies. What are the antibodies? Antibody and B. If these antibodies are present, these antibodies will react with the blood. Okay, it is absent in the AB blood group, that's why it is universal residue. Following are the stages of pathway of conduction of an action potential. So action potential, where it is originating. Okay, SA node, auto excitable, that's why our heart is known as myogenic heart. SA node is the pacemaker, so electrical impulse is generating here. After SA node, it will go to AV node. AV node. From the AV node, it will go to AV bundle. From AV bundle, it will branch bundle of His. Then it will spread along the ventricles. That is Purkinje fibers. So this is the order: SA node, AV node, AV bundle, bundle of His, Purkinje fibers. Now the question is related to ECG. ECG. P wave. What does a P wave indicate? P wave, different meanings are there. One meaning is depolarization of atria. Depolarization of atria, generation of action potential, okay, contraction of atria, both atria. So these all are indicated by P wave. Electrical excitation in the SA node, depolarization of atria, contraction of the atria or atrial systole. Okay, QRS complex, depolarization of ventricle and contraction systole, ventricular systole. T wave, T wave is the repolarization of ventricle. End of T wave indicates end of T wave. What does it end of T wave indicates? End of T wave indicates end of ventricular systole. Ventricular systole. End of T wave is indicating the end of ventricular systole. So here T wave is this. TP gap, Heart muscles are electrically silent. So here it will be in a, all the heart chambers are silent means which phase? Joint test. What would be the heart rate of a person if the cardiac output is given? Cardiac output is 5 liter of 5 liter blood volume in the ventricles at the end of diastole, means end diastolic volume at the end of diastole 100 ml. Clear? At the end of systole of ventricle, end systolic volume 50 ml. From here we can calculate the stroke volume. Stroke volume is equal to end diastolic volume minus end systolic volume, that is 50 ml. Okay, 50 ml. So cardiac output we can mention as stroke volume into heartbeat. So stroke cardiac output is given 5000 ml. Stroke volume is given 50 ml. So from here we can calculate the heartbeat. 100 beats per minute. 100 beats per minute. 100 beats per minute is the things. Next one, nitrogenous waste is excreted in the form of pellet or paste. So in the form of pellet or paste, who will excrete? Birds. Okay, birds. This is fishes, amphibia, mammals. So A options. Pavocrystatus. Which of the following statement are correct? Excessive loss of body fluid from the body. Switch off osmoreceptor. If the body fluid is, if the loss of body fluid takes place, it has to switch on, not the switch off. So this is wrong. ADH facilitates water reabsorption. Water reabsorption will take place from the collecting duct and in the DCT. This is correct. ANF causes vasodilation. That is correct. ADH causes increase in blood pressure. That is also correct. ADH is responsible for decreasing. It will not decrease the, it will increase. Clear? C, B, C, D. B, C, D is the correct. Arrange the events. Renin-angiotensin mechanism. Renin-angiotensin-aldosterone system. So when it will operate? Fall in blood pressure. Fall in blood pressure. If the blood pressure decreases, it will activate the JG cells to release renin. Okay. What this renin will do? Renin will convert the angiotensinogen into angiotensin 1. So this is the function of renin. Angiotensinogen into angiotensin 1. Angiotensin 1 to angiotensin 2. This conversion is angiotensin converting enzyme. This is present in the lungs. Clear? After this angiotensin is a vasoconstrictor. Angiotensin is vasoconstrictor. It will constrict and also it will activate the adrenal gland. So aldosterone will be released. 1, 2, 3. Aldosterone will be released. If the aldosterone is released, it helps in the reabsorption of sodium and water from distal convolution. Reabsorption of sodium and water will take place. Clear? So this is the order. Next one. Calcium is important in skeletal muscle contraction because so in troponin there is a region known as calcium binding region. Okay, if calcium bind to troponin, remove the masking effect. If the masking effect is removed, so that crossbridge formation takes place. Okay, calcium is important in skeletal muscle for contraction because it will bind to the troponin to remove the masking of active site on actin. So this is the correct one. Activates myosin ATPase, not correct, detaches the myosin head from the actin. So for the cross bridge breaking, ATP is required. Prevent the formation of bonds between the myosin. That's also not. Floating ribs. Floating ribs, there are three. True ribs are there, true ribs. First 7 pairs are true ribs. First 7 pairs. 7 pairs are true ribs. Then 8, 9, 10. 8, 9, 10. These are called false ribs. 11 and 12, that is floating ribs. Floating ribs, you can see this is floating. Do not connect with the sternum. It is not connected with the sternum. Directly also not connected, or it is not connected to the cartilage also. That is why floating ribs. Acromion process, acromion and clavicle. Clavicle is connected to acromion and sternum. So clavicle is the collarbone. Scapula. Scapula is located between the second and seventh ribs. This is the correct one. Then glenoid cavity. In the glenoid cavity, humerus, head of the humerus will articulate. So this is the correct matching. Out of X pairs of ribs, how many pairs are there? There are 12 pairs. X pairs means there are 12 pairs of ribs are there in human bird. Only Y pairs are true ribs. How many pairs? 7 pairs are true ribs. So Y is 7. 7 pairs are true ribs. Okay, select the option that correctly represent the values of X and Y and provides their explanation. What is the explanation? Two ribs are attached dorsally to the vertebral column, because vertebral column is dorsal, ventrally to the sternum. Okay, that's correct. This is the correct one. Next one. Fibrous joint. Fibrous joints are immovable joints. Immovable joint is present in the skull, and tooth is located in the jawbone, socket of the jawbone. So that is also immovable joint. So this is immovable. Cartilaginous joint is present in the vertebrae, not in the between adjacent vertebrae. What is present? Limited movement. That is cartilaginous joint. Cartilaginous joint. Except condyloid and pivot joint. Clear? Hinge joint. Which is the hinge joint? Knee. Help in locomotion. Hinge joint. Ball and socket joint. Humerus and pectoral girdle. Ball and socket joint. Humerus and pectoral girdle in rotational movement. Ball and socket joint. Select the correct matching of the type of joint with the example. Gliding joint. Gliding joint is present in the carpals, okay, and tarsals. So carpals, this is correct. Cartilaginous joint is not in the frontal and parietal. Here it is immovable joint. Pivot joint, radius ulna pivot joint. First and second vertebrae pivot joint. Hinge joint, knee joint. Okay. So this is also not correct. Which of the following are autoimmune disorders? Autoimmune disorders means our immune system cannot distinguish between the foreign cells and the self cells. So they will attack the self cells. That is autoimmune response. Myasthenia gravis. Myasthenia gravis, antibodies attacking the receptor of acetylcholine in neuromuscular junction. So this will happen. Myasthenia gravis, paralysis. Rheumatoid arthritis, yes. Gout is the accumulation of uric acid crystals in joints. Muscular dystrophy is the genetic disorder, because here dystrophin protein is not formed. So don't confuse with myasthenia gravis and muscular dystrophy. This is genetic disorder. This is what? This is autoimmune disorder. Systemic lupus erythematosus. This is also autoimmune disorder. So these three things are autoimmune. The remaining are not. Chronic autoimmune disorder affecting the neuromuscular junction, leading to fatigue, weakening, and paralysis of skeletal muscles. Skeletal muscles will get paralyzed. Myasthenia gravis. Okay. So muscular dystrophy, progressive degeneration of skeletal muscles. Dystrophin protein is not there. Gout, uric acid accumulation. Arthritis, inflammation in joints. Now match the following. Gout. What is gout here? Gout is accumulation of uric acid crystals in the joints. Osteoporosis. Osteoporosis will take place if, in the case of females, if the estrogen level is low, then demineralization of the bone takes place. That we call it as osteoporosis. So this is osteoporosis due to decreased level of estrogen. Tetany, wild spasm due to low calcium in the, if the low calcium is there, what will happen? Sodium channel will always open. So continuous depolarization takes place. This is the one. Tetany. Muscular dystrophy. Muscular dystrophy is a genetic disorder. Muscular dystrophy is genetic disorder. Next one, match the following. List 1 and list 2. Pons. Pons, what is the function of pons? Pons connect different parts of the brain, and also pneumotaxic center is present in the hypothalamus. Hypothalamus contain neurosecretory cells. So hypothalamus can produce both inhibitory and releasing hormones. Medulla oblongata. Medulla oblongata is involved in controlled respiration and gastric secretion. Cerebellum provides additional space for the regulation and posture and balance. So here the different parts of the brain and their function, very very important part. The part of human brain that helps in regulation of sexual behavior, expression of excitement, pleasure, rage, fear. So these all are controlled by limbic system. So limbic system parts you should know: limbic system, amygdala, hippocampus, hypothalamus, olfactory lobe. These all are the parts of limbic system. So limbic system is controlling the fear, rage, pleasure, excitement, expression, motivation. So these all are under the control of limbic system. Brain stem of human brain consists of midbrain. And in hindbrain, what are the parts? Pons and medulla. Cerebellum is not there. So this combinedly we call it as brainstem. Now the function of parathyroid hormone. Parathyroid hormone is very important. Parathyroid hormone and thyrocalcitonin is jointly regulating the calcium level. So here parathyroid hormone increase the blood calcium. Thyrocalcitonin will decrease the blood calcium. Stimulate the process of bone resorption. It will stimulate the process of bone resorption. That is correct. But the question is not the effect of parathyroid, which is not the effect. Decrease in blood, it is not the effect. Resorption, reabsorption of calcium by renal, that is the function. Decrease the absorption of calcium, increase the absorption of calcium from digested food. Increase metabolism, decrease absorption of calcium from digested food. So it will increase the absorption, not decrease. Increase metabolism, it will not increase the metabolism. So these are the correct one. Stimulate process of bone resorption, that is correct. Decrease calcium level in blood, it will increase the calcium level in blood. Decrease the absorption of calcium, so it will increase the absorption. Okay, so these two things are the correct one. Graves disease is due to, Graves disease also known as exophthalmic gout, hypersecretion of thyroid. A pregnant female delivers a baby who suffers from stunted growth, mental retardation, low intelligence quotient, and abnormal sin. This is due to some problems. Okay, this is due to the deficiency of iodine. If iodine deficiency is there, thyroid gland is not functioning properly. So these are the problems the newborn baby will experience. Amino acid tryptophan. What is tryptophan? Tryptophan is a complex amino acid. Most complex amino acid. Most complex amino acid. Most complex amino acid. That is tryptophan. And also tryptophan is most complex amino acid, and it is also aromatic one. It will produce melatonin and serotonin. Melatonin and serotonin is produced from tryptophan. Which of the following is not the function of placenta? Not the function of placenta. Secretes oxytocin. Oxytocin during parturition. That is not the function of placenta. Oxytocin is not at all secreted by placenta. Facilitate supply of oxygen and nutrients, that's correct. Estrogen, correct. Removal of carbon dioxide waste material. So function of placenta, focus on the function of placenta and the hormones secreted by the placenta. So what are the hormones secreted by the placenta is very important during the pregnancy and during parturition. Which of the following is not a steroid hormone? Hormones are steroid hormone, protein hormone, amino acid derivative. Okay. Progesterone is steroid, glucagon is peptide, cortisol is steroid, testosterone is also steroid. So this is not steroid hormone. Steroid hormone can directly enter inside the cell. The receptors will be present on the surface of the nuclear membrane, and also it is present in the cytoplasm. Select the correct sequence of transport of sperm cells in male reproductive system. So in male reproductive system, starting from testis. So inside the testis, what is there? Seminiferous tubule is there. From seminiferous tubule, it goes to rete testis, vasa efferentia, epididymis. So epididymis is a structure where the storage of sperm takes place, functional maturation of sperm takes place. Then it goes to vas deferens, ejaculatory duct, urethra, and urethral meatus. Next one, arrange the component of mammary gland from proximal to distal. So which is the first one? Mammary gland milk production takes place in alveoli. So from the alveoli it goes to mammary tubule. From the mammary tubule it goes to mammary duct. From the mammary duct, mammary ampullae. From the mammary ampullae, lactiferous duct. So this pathway is very, all type of pathway, sperm movement, ovum movement, milk production, secretion, and their discharge. These things are very important. How many secondary spermatocytes are required to form 400 million spermatozoa? From one secondary spermatocytes, two spermatid is forming. Two spermatid formation takes place. Consider this is a secondary spermatocyte. Secondary spermatocyte. From the secondary spermatocyte, this is what secondary spermatocyte is haploid. So from one secondary spermatocyte, two haploid spermatid is forming. Spermatid forms spermatozoa. So here maturation takes place, spermatozoa. Clear? So here if it is 200 million, then spermatozoa formation will be 400 million. So the point I have to remember is secondary spermatocyte is haploid, primary spermatocyte is diploid, spermatogonium is diploid, spermatid is haploid, spermatozoa is also haploid. What is the correct sequence of sperm formation? Sperm formation takes place, okay, in testis. In the testis, seminiferous tubule contain two types of cells. One is spermatogonium. From the spermatogonia, spermatocyte is forming. This is diploid. Spermatocyte first, it will form primary spermatocyte, then secondary spermatocyte. Secondary spermatocyte is haploid, primary spermatocyte is diploid. Then it will form spermatid. Up to spermatid, we will say the process is spermatocytogenesis. From that, spermatid is maturing into sperm. Spermatid to sperm. This process we call it as spermiogenesis. So for this process, Sertoli cells is required. They will produce some factors for the maturation of sperm. So this is the correct one. Which of the following hormones will cause a release of ovum from Graafian follicle? So in the mid of menstrual cycle, there will be LH surge. LH hormone will increase. Due to sudden increase in the LH hormone, Graafian follicle will rupture. Rupturing of the matured follicle will take place. So at the same time, there will be two other hormones that also will be high. FSH is also high, and also estrogen also will be high. These three hormones levels will be high during the mid of menstrual cycle. leads to the breakdown of Graafian follicle, and it will form a temporary endocrine gland that we call it as corpus luteum. And this corpus luteum will produce hormone called progesterone. Progesterone will maintain the pregnancy. So here, which of the following hormone level will cause release of ovum, ovulation from the Graafian follicle? High concentration of progesterone? No. Low concentration of LH? High concentration of LH is required. Low concentration of FSH? No. High concentration of estrogen. So here estrogen is not directly mentioned. During the mid of menstrual cycle, these hormones level will be high. So one of that hormone is estrogen. So we can go for the estrogen. Human beings at the end of 12 weeks, means first trimester, if completed, what will happen? Most of the major organs will be formed. Head of this is forming in the 5th month. Movement takes place in 5th month. So most of the organs will be formed in the first trimester, that's the 12 weeks. Hormones secreted by the placenta to maintain the pregnancy. To maintain pregnancy, what is required? Maintain pregnancy, all the hormones of placenta is required. For example, human chorionic gonadotropin, human placental lactogen, estrogen is required. Prolactin is not required. This is not required. Relaxin is not required. Relaxin is required for parturition. Glucocorticoid is also not required. So here, prostrogen, estrogen, HPL, human chorionic gonadotropin. Which of the following is not an important component of initiation of parturition? Parturition initiation means birth process. Oxytocin should be released so that contraction will take place. Prolactin is required for milk production, not for parturition. Increase in estrogen progesterone ratio. Estrogen progesterone ratio will increase, means progesterone level will decrease. That leads to parturition. Synthesis of prostaglandin is required for contraction. So prolactin is not required. Now related to the birth control measures, non-mediated IUD. So non-mediated IUD also we call it as inert IUD. So it will trigger the inflammatory response. Non-mediated IUD is lips loop. Lips loop is non-medicated IUD. Copper releasing IUD, multi-load 375. Copper releasing. Hormone releasing IUD, which is the hormone releasing LNG 20. Hormone releasing IUD. Implants containing progestogen. Correct. So this is the correct one. Next one. Which of the following is not an intrauterine device? Intrauterine device and their function. It will increase the phagocytic process. Okay. If the progesterone intrauterine contraceptive measure is used, so progesterone will thicken the cervical mucus. So sperm entry will be retarded. Progestasert is LNG20 and progestasert are hormone releasing. That is IUD. Progestogen is used in pills and in implants. Okay. Multi-load 375, lips loop, these all are intrauterine contraceptive device. Progestogen is just a hormone. List 1, list 2 with respect to the method of contraception. Diaphragm. So diaphragm is involved in barrier method, cover the cervix, blocking the entry. Okay, but diaphragm cannot prevent STDs. Contraceptive pills. Pills is acting on the release of GnRH, gonadotropin releasing hormone. So what will happen if it is not released? LHFSH also will not release. So what will happen? Inhibit the ovulation and implantation process. Intrauterine device IUDs increase the phagocytosis of sperm. Lactational amenorrhea means during lactation period there is no menstrual cycle, so at that time gonadotropin will not be released. So what will happen? Absence of menstrual cycle. This is the correct one. Contraceptive pill Saheli. Saheli is the first non-steroid pill. There is no steroid, so side effect is less. Non-steroid pill, and it is a weekly pill, and there is a chemical known as sencromen. Sencromen is a chemical present in this pill. Developed in Lucknow, Central Drug Research Institute, Lucknow. So here how it is acting? It is acting on the estrogen receptors. So block the estrogen receptors. If the estrogen receptors are blocked, what will happen? Estrogen is required for the thickening of the endometrium. So that process will not take place. If the endometrium is not thickened, implantation also will not take place. So estrogen receptor is blocked. Estrogen action is not there. Consider this is estrogen. Receptors are blocked, so action is not there. Thickening of endometrium is not there. Then it will act. It will inhibit the process of implant. Without following sexually transmitted infectious, completely curable. HIV is not curable. Syphilis, trichomoniasis, this is curable. Hepatitis B, genital herpes. Genital herpes is not cured. Genital warts is cured. Genital warts is curable. Herpes is not cured. Next one. In case of couple where the male having low sperm count, if the sperm count is low, the procedure one has to follow means artificial insemination. Artificial insemination or intrauterine insemination. If semen is inserted in this vagina, we will call it as artificial insemination, and if it is in the uterus, intrauterine insemination. If the sperm count is too low, then this is more preferred. Without following is not an autoimmune disorder. Autoimmune disorder means the immune system is attacking its own. Alzheimer's. Next one. Cytokine barrier. Cytokine barrier are virus infected cells will secrete some cytokines that's called interferons. This will not allow the virus to attack the other cell, neighboring cells, because in these cells, neighboring cells will produce translational inhibiting proteins. It will inhibit translation of viral proteins. So viral proteins will not form. Okay. Cellular barriers, neutrophils and macrophages. Cellular barrier. Phagocytosis. Physiological barrier, tears and saliva is acting as physiological barrier. Physical barrier, skin is physical barrier, mucus coating, respiratory tract. Without following reason is mainly responsible for graft rejection. So we have two types of immunity: cell mediated immunity and humoral immunity. So cell mediated immunity is responsible for graft rejection. That is direct question. Select the incorrect statement with respect to acquired immunity, which is incorrect. Acquired immunity means the immunity the person acquiring during the lifetime. That is specific to a pathogen. Acquired immunity is non-specific. It is specific. Defense at the time of birth. At the time of death, birth means it is congenital. After the birth, the person is acquiring, that is acquired immunity. So this is the incorrect one. Primary immune response is produced by our body encounters. Primary immune response is the first immune response. During first immune response, IgM antibodies will be high compared to IgG. Secondary immune response. In secondary immune response, IgG will be high compared to IgM. Okay. So this point you have to remember the antibody production during the immune response. Colostrum, yellowish fluid secreted by the mother during the initial days of lactation. Colostrum contains which antibody? Colostrum contains IgA antibody. Body secretions mainly contains Ig. Saliva contains Ig antibody. Tears contain IgA antibody. IgG can cross the placental barrier, which is the smallest antibody. Antivenom injection contain preformed antibody, means serum contains preformed antibody which is produced in other animals, while polio drops are administered into the body. Polio drops contains inactivated pathogen. Inactivated, attenuated or weakened pathogen, but it is considered as live vaccine. if it is containing inactivated pathogen or attenuated pathogen, weakened pathogen, considered as live vaccine, and it is the first generation vaccine. So in first generation vaccine, the complete organism is used for making the vaccine. Which of the following is the correct regarding AIDS causative agent? AIDS causative HIV virus, human immunodeficiency virus, and it is a retrovirus. Retrovirus means this RNA can form complementary DNA. Complementary DNA is found from RNA using reverse transcriptase. So two molecules of reverse transcriptase enzyme is there, two molecules of single stranded RNA is there. So in the case of this RNA virus, this RNA virus is what? This is retrovirus. Retrovirus. And this retrovirus means this RNA is forming the complementary DNA. That's why retrovirus. And the enzymes are present here. What are the enzymes present? Enzymes present in this virus. One is reverse transcriptase. Reverse transcriptase. Reverse transcriptase enzyme is used for reverse transcription, RNA to complementary DNA. Next one is protease. Protease is also present for breaking the capsid. Protease is present. Then viral DNA has to be integrated with the host DNA. For that, integrase enzyme is also there. So these all are the enzymes present in the HIV virus. Cocaine. What is cocaine? Cocaine is a stimulant. Cocaine will interfere with the neurotransmitter dopamine. It will interfere with the neurotransmitter dopamine. So here cocaine is obtained from erythroxylum coca. Heroin. Heroin is produced from morphine by acetylation. Acetylation of the morphine will give heroin. It is from Papaver somniferum. Morphine is a sedative. Morphine is a sedative. If the person is taking the morphine after the surgery, all the body functions will get reduced. So it's a sedative, it is a painkiller. It will not induce the sleeping process. That is morphine. Marijuana. Marijuana is produced from cannabis sativa, and this marijuana is produced from the dried leaf, not dried leaf, dried flower of cannabis. In this dried flower, there is an active ingredient which is giving the effect, that we call it as delta 9 THC, delta 9 tetrahydrocannabinoid. So this ingredient is isolating and this ingredient is giving the effect. So marijuana is produced from in this way. Which of the following is drug obtained from the plant? Flower show. So this is datura. Datura. This flower is datura, and datura is hallucinogen. So what are the other types of hallucinogens? One is datura, Atropa belladonna, LSD, lysergic acid diethylamide. So lysergic acid diethylamide is synthetic. It is obtained from the fungus Monascus purpureus. Next one, psilocybin. Psilocybin is also giving the hallucinogenic effect. So psilocybin is called magic mushroom. Magic mushroom. Magic mushroom is psilocybin. Then mescaline. Mescaline. These all are the hallucinogenic. These all will give the hallucinogenic effect. Datura, Atropa belladonna, LSD, psilocybin, mescal. Which of the following method is not commonly used for introducing DNA into the plant cell? For introduction to the plant cell, we are using the agrobacterium mediated. transformation, gene gun, gold particle coated with the DNA, disarmed pathogen. But bacteriophage we are not using. Bacteriophage is for bacteria. Which of the following restriction site in E. coli? PBR X and Y genes. So here you can see BAM H1 Sal N1. Which site is there here? Tetracycline. Tetracycline site is there. Tetracycline. Tetracycline. Okay. Tetracycline resistant. Here ampicillin resistant. Ampicillin resistant site is there where PVU1 and PST1 having restriction site. BAM H1 and SAL1 having the restriction site in tetracycline resistant. Clear? So if you are removing this part, this will not show the resistance against tetracycline. If you are removing this part and inserting other gene, they will not show the resistance against ampicillin. So the question is regarding X and Y. So X is regarding the ORI site, X is ORI site, origin of replication, ORI site. This is required for replication process. Then here Y is mentioned here. What is Y here? Y is raw protein. This protein is required for replication of the plasmid. Ligation of foreign DNA at which of the following site will result in loss of tetracycline. So tetracycline resistant we can see here. You can see the BAM H1 here, tetracycline. Now if you are removing that with the help of BAM H1, then it will lose the tetracycline. So enzyme are required to cut the site, that is BAM H1. Which of the following is not desirable feature of a cloning vector? So one cloning vector, if the cloning vector have more than 2 or 3 recognition site for a particular enzyme, then it will complicate. So we will get a lot of fragments. More fragments will be generated. This is the complication. More fragments will be generated. If more fragments are generating, it will complicate the cloning process. Plasmid PBR322 has PST1 restriction enzyme site with gene ampicillin resistant. Gene ampicillin resistant that confers ampicillin resistance if this enzyme is used for inserting a gene beta galactosidase. So this enzyme we are using for inserting this gene by removing the ampicillin resistant site. If the ampicillin resistant site is removed with the help of PST1 and inserting another gene beta galactosidase, so what will happen? In case of substrate, it will produce color, but it will not give the resistance against ampicillin. Resistance against ampicillin will not be there because already we are removing the ampicillin resistant gene with the help of PST1. In that place, we are inserting beta galactosidase. Okay, so beta galactosidase will be active if you are treating with a substrate. So we will get colored product, blue color. The introduction of tDNA into the plasmid. How? Infection of the plant by Agrobacterium tumefaciens. So Agrobacterium tumefaciens contain tDNA. Transfer DNA. Which of the following technique does not serve the purpose of early diagnosis disease? Early diagnosis, traditional method we cannot use for early diagnosis. All the recombinant DNA technology, enzyme linked immunosorbent assay, this we can use. The principle is antigen antibody interaction. Antigen antibody interaction. So this is the principle, antigen antibody interaction. Recombinant DNA technology, serum urine analysis. Serum urine analysis is the traditional method. Using this, we cannot identify. Select the incorrect statement regarding the chemical structure of insulin. Chemical structure of insulin. Mature insulin molecule consists of 3 peptides ABC. So that is not correct. Only A and B will be there. It will be joined together with the help of disulfide. In the case of immature one, extra C peptide will be there. C peptide will be removed in the mature one. So here the answer, this is correct. Gene gun used for transferring the gene in plants. This is used for transferring the gene in plants. Gene therapy used for correcting the gene. Gene cloning. Using gene cloning, we can multiply. More copies we can produce. Gene cloning. Genome is the total DNA content in the organism. So these are the match the following simple questions. Only you should know that what is therapy, gene therapy. What are the techniques used for transferring in plants, transferring in fungus, transferring in animals. In animals, we are using the retrovirus. So retrovirus are specialized for animal cell. Gene therapy, adenosine deaminase. If the enzyme is not produced, B and T lymphocyte will not mature and it will lead to severe combined immunodisorder. So this disease is very complicated disease. Immune system failure takes place. So for this, we can use genetically engineered lymphocytes, but there is a problem in this. These lymphocytes are not immortal. That is the problem. Retrovector introduced these lymphocyte gene isolated from bone marrow producing ADA is introduced into the cell in the embryonic stage. So in early embryonic stage, this is a permanent cure. So the remaining all are not permanent cure. Why this genetically engineered lymphocyte we cannot consider as a permanent cure? Because they are not immortal cells. After their lifespan, they will die. With regard to insulin, choose the correct one. Which is the correctness? C peptide is not present in mature. That is correct. Insulin produced by recombinant DNA technology has C peptide. C peptide is not present. C peptide will be removed. Proinsulin has C peptide. Proinsulin has C peptide. Peptide, B peptide of insulin are interconnected by this. So this is also correct. A peptide and B peptide are interconnected by disulfide bridge. Proinsulin has C peptide. That is correct. Okay. So these are the correction. Next one. BT cotton. BT cotton is Bacillus thuringiensis. Deficiency of adenosine. Deficiency of adenosine deaminase. Adenosine deaminase deficiency we can cure using gene therapy. RNA interference is the cellular defense in eukaryotes. So this is the correct one. PCR is used for amplification of genetic material. So you can detect the pathogen. So HIV infection. Which of the following is commonly used vector for introducing DNA fragment in human lymphocyte? So in human cells, the vector we are using is retrovirus. Retrovirus are specially for animal cells. So retrovirus we are using. Alpha 1 antitrypsin. Alpha 1 antitrypsin is used for treating emphysema. Direct question. Cry1AB, Cry2AB, Cry1AC, Cry2AB. So Cry1AB is particularly for corn borer. Cry1AC is for cotton bollworm. Enzyme replacement therapy. So enzymes are provided from external source, that is ADA deficiency for treatment but it is not the permanent cure. Alpha antitrypsin, emphysema, Cry1AB, what is Cry1A? Cry1AB in corn borer. Cry1AC, that is cotton boll. Which body of the government of India regulates GM research, genetically modified, and safety of introducing GM organism, genetically modified organism for the. So there So there is an organization which is controlling this, that is Genetic Engineering Approval Committee. Genetic Engineering Approval Committee is monitoring the ethicality of this research. So if the research is going on, is it ethical, is there any benefit for the human being? So these all are monitored by Genetic Engineering Approval Committee. So approval from this committee is required for doing any research in this biotechnology, or anything we are modifying the genes. Okay, if we are producing the genetically modified organism, for that we need the approval, because we are altering the genome of an organism, so it can leads to some complication. So there are some limitation for doing this work. After getting the permission from this committee, this work is carrying. So these are the special area where you have to focus. In biotechnology, what you have to focus in biotechnology, the vector, which vector is using, and enzymes, functions. Okay, so these are the basically all area we covered, from starting from the tissues. So every chapter there are specific area where you have to focus. So that area, read in depth, analyze the points where you will get the question for 100%. For example, respiratory volumes and capacities, pressure difference. So these are the area you can expect from the respiration. One question will be sure. Okay.